Welcome to this deep dive. Today, we're pulling from field biology journals, indigenous science, and ecological data for a really specific mission.
We are. We're investigating the lifelong dialogue between DNA and the world around it.
Right. And we are stepping into a very specific narrative frame today. You, the listener, are officially stepping into the role of a Bioforge growth investigator.
Which is a pretty critical mission, honestly. Your overarching goal here is to study why organisms grow differently in different environments.
Yeah, and by completing that mission, you'll earn the MS-LS1-5 investigator badge.
Exactly. But to secure that badge, you have to master a core learning target.
Right, which is basically, I can use evidence to explain how genetic factors and environmental conditions work together to influence how organisms grow.
And we should probably pause and define our terms real quick, right? Like, an organism is just any living thing.
Yeah, from a giant redwood tree down to the bacteria in your gut.
Right. And growth is just how they get bigger or develop over time.
So think of it like a towering skyscraper. You wouldn't just look at a massive building and assume it, magically appeared.
No, obviously not. You know there's a blueprint somewhere. A highly detailed document dictating where every single steel beam is supposed to go.
And here's the catch. If the soil on the building site is too soft or, I don't know, there's a massive concrete shortage or something.
Yeah, the final skyscraper is not going to perfectly match that original blueprint.
Exactly. The environment on the ground dictates how the architect's instructions actually get executed.
And that is the exact dynamic we're applying to living things today.
Because in science, we can't just look at a tree getting bigger and guess why it happened. We need an explanation backed by solid observation.
We have to figure out what caused that growth. So we start with the biological blueprint. Every organism receives an internal set of instructions from its parents.
Which are inherited traits, right?
That's exactly. Inherited traits. They are encoded on chromosomes and act as these regulatory templates.
Which brings us to our first major concept for the badge, which is genetic factors.
Right. A genetic factor is a specific piece of that inherited code that influences how an organism develops.
But we need to clarify something right out of the gate here. Genetic factors set an organism's growth potential.
Yes, they set the ceiling. They do not offer a strict guarantee.
Setting the potential is such a crucial concept. Let's look at a pretty dramatic example in agriculture. Belgian blue cattle.
Oh, yeah. Those things are wild.
Right. They have this specific inherited mutation in a gene that produces a protein called myostatin.
And normally, myostatin is basically a biological stop sign for muscle growth.
Yeah.
Tells the body, okay, we have enough muscle, you can stop building now.
So the Belgian blue cattle basically just have a broken stop sign.
Precisely. Because of that specific genetic factor, their biological potential for muscle growth is just vastly higher than smaller breeds of cattle.
That's incredible. Or, even stepping away from animals, consider plant seeds.
Right, like sunflowers.
Yeah. If you plant seeds from a giant sunflower variety and a dwarf sunflower variety in the exact same soil, they grow at fundamentally different rates.
Because their internal blueprints establish completely different ceilings for their growth. Even in wildlife, we see this. Look at the Sitka black-tailed deer.
Oh, sure. They inherit very specific genetic traits that dictate their baseline size, their coat color, all of that.
Right. But that raises a really massive question for our Bioforge investigation here.
Which is, if a plant has the genes to be 10 feet tall, or a deer has the genes to weigh a certain amount, is that blueprint a strict promise?
Does it always reach its genetic potential?
Right. Is it guaranteed to hit 10 feet no matter what?
And the answer is it rarely does.
Mm-hmm.
Because the blueprint never operates in a vacuum. The environment is what actually manages the construction site.
Which introduces our next big concept. Environmental conditions or environmental factors.
Exactly. These are the external realities surrounding an organism. And they absolutely dictate how that genetic potential actually gets expressed in the real world.
We're talking about variables here.
Right, basically anything that can change and affect the outcome.
And the list of environmental factors is huge, right? I mean, food availability, water, light, temperature.
Visual, nutrients, physical space, the size of the habitat.
Just general resource availability, yeah. All those variables physically limit or accelerate the blueprint.
Think about a severe drought. You might have a crop with a genetic potential to yield this massive harvest.
But if there's no water?
Exactly. A lack of water forces the plant to conserve energy. It stops building new tissue, which drastically reduces its actual growth.
But on the flip side, If you introduce an environmental variable like a nutrient-dense fertilizer, you see a massive spike in growth.
Right.
But I want to dig into the actual mechanism of that interaction for a second, because isn't there a physical mechanism where the environment essentially reaches in and flips genetic switches?
Yeah, you're talking about epigenetics.
Yes, I've
Yep.
read a bit about epigenetics, and it feels highly relevant here.
It is the perfect mechanism to explain this. Epigenetics refers to changes in how genes are expressed without actually altering the underlying DNA code itself.
So the blueprint stays the same, but how it's read changes.
Right. So when an organism encounters environmental stress, let's say an Arctic plant facing a really short summer with very limited light.
Right, because of where it is on the globe.
Exactly. The environment actually triggers chemical modifications. It attaches these molecular tags to the DNA that literally turn certain growth genes off or on.
Wait, so the Arctic plant remains short and small not just because it's cold, but because the lack of light chemically instructed the plant's genome to stop growing up.
Yep. It tells it to prioritize survival over upward growth. That is a biological reality of it. And
Wow.
we see this with aquatic life, too. Habitat size is a huge environmental factor.
Oh, like fish in a pond.
Exactly. Fish growing in small, confined ponds often max out at much smaller sizes than the exact same species living in large lakes.
And water temperature plays a role too, right?
Yeah.
And salinity.
Huge role. Cold water holds more oxygen, but it slows down a fish's metabolism.
Yeah.
That radically alters how their genetic growth potential plays out compared to if they were in warmer waters.
And even large mammals aren't immune to this. Bear sizes fluctuate wildly based on food availability and their specific habitat conditions.
Oh, for sure. A bear with an abundance of high-fat salmon in its territory is going to express a much larger physical size than a bear with the exact same genetic potential living in a scarce environment.
Which brings us to a really mandatory misconception check for our Bioforge investigators.
We have to state this as clearly as possible.
Yes, genes do not act alone, and the environment does not act alone.
Growth usually reflects both inherited factors and environmental conditions.
Right. It's a lifelong dialogue between the internal blueprint and the external reality. You just cannot separate them.
It's an ongoing, totally dynamic interaction from the moment an organism begins to develop all the way until the end of its life.
So, to see this lifelong dialogue in action, our sources point us toward this massive real-world laboratory, basically, the state of Alaska.
Oh, yeah. The environmental variables there are pushed to absolute extremes.
You have these dramatic seasonal changes, intense fluctuations in daylight patterns.
Freezing water temperatures, massive snow cover, really localized food availability.
Right. And local observers, indigenous elders, subsistence users, fishers, gardeners, field biologists, they've all tracked these variables for generations.
They constantly monitor how those extreme seasonal changes affect growth patterns, from the size of berries in the summer to the density of offshore kelp forests.
But looking at the field data in our sources, there was one phenomenon that initially confused me.
Oh, the salmon trees.
Yes, the forests of Alaska, specifically the Sitka spruce tree and spawning salmon.
It's so fascinating.
The data says these massive terrestrial trees get up to 70% of their nitrogen directly from salmon.
Yep, 70%.
But trees obviously don't eat fish. I mean, are we talking about the soil simply absorbing the decay, or is there a more complex mechanism happening here?
It is a brilliantly complex mechanism. It's known as a marine nutrient subsidy.
Okay, break that down for me.
So, when salmon swim up the rivers to spawn, predators like bears and eagles pull millions of pounds of fish out of the water and into the surrounding forest.
Right, because they hunt them.
Exactly. They consume the calorie-dense parts and just leave the rest of the carcasses on the forest floor.
Delivering marine biomass directly into a terrestrial biome.
And then the microscopic environment takes over. Bacteria, maggots, and highly specialized mycorrhizal fungi.
Which are basically those fungal networks attached to the tree roots.
Right, exactly. They break down those decaying fish. And the salmon are incredibly rich in a specific environmental nutrient, which is nitrogen.
Ah.
The fungal network absorbs this nitrogen and literally pumps it directly into the roots of the Sitka spruce.
Wait, so the trees are basically being fertilized by fish. It acts as this incredibly potent, naturally occurring fertilizer.
It really does, and it radically alters the tree's growth rate. Field biologists actually use stable isotope analysis to track the origin of the nitrogen inside the tree rings.
Which proves it comes from the ocean. That's amazing.
Yeah. Because of this sudden, massive influx of high-quality nutrients, the genetic potential of those spruce trees is supercharged. The cellular division in the tree just accelerates.
And the numbers in the study are staggering. Let me look at this. A Sitka spruce growing adjacent to one of these salmon streams takes only 86 years to reach a trunk thickness of 50 centimeters.
Right. Now compare that to a spruce with the exact same genetic blueprint, but growing along a river that does not have a salmon run.
How long does that one take?
That tree takes 300 years to reach the exact same 50-centimeter size.
Wow. 86 years versus 300 years?
Yep, the genetic blueprint is identical, but the environmental variable, the salmon-derived nitrogen, it basically hits the biological accelerator.
It's like early farmers were playing a game of genetic matchmaking without knowing the actual rules of genetics,
No, exactly. Science is fundamentally a human endeavor of noticing and utilizing these exact patterns. Long before we had stable isotope analysis or even the vocabulary to discuss chromosomes, early agriculturalists used selective breeding,
Right. They observed how environmental conditions and inherited traits affected growth.
using trial and error in plant breeding and animal husbandry to manipulate the outcomes. They couldn't see the DNA code, but they looked at the final physical traits.
Like drought-resistant wheat or heavily muscled draft horses.
Right, and they tried to stack the deck in their favor by selecting which organisms reproduced. And we see incredibly sophisticated examples of this in indigenous science and traditional ecological knowledge.
I read about this in the materials, the Tlingit engineering in Southeast Alaska.
Yeah, the stone weirs.
Right, these underwater walls and traps for fish that date back over 11,000 years. But I'm curious about the specific mechanics here. How does building a stone wall in a river translate to managing genetic and environmental growth factors?
It comes down to socio-ecological optimization.
Okay.
These weirs weren't just blunt instruments designed to catch every single fish. They were engineered to act as selective filters.
Oh, interesting.
By adjusting the gaps in the stones and managing the timing of the harvest, the Tlingit could selectively harvest certain fish while intentionally allowing others to pass upstream to spawn.
Which were probably the largest, healthiest individuals.
Exactly. So they were actively shaping the inherited genetic potential of future generations of salmon.
While simultaneously ensuring the environmental variable, meaning the total population size, remained balanced enough to not exhaust the river's resources.
Right. They utilized this incredibly deep generational understanding of cause and effect in nature. Our modern methods evolved from these early field observations to the systematic testing by scholars like Ibn al-Haytham in the 10th century, all the way to our modern controlled experiments.
Yet the foundational goal is totally the same, uncovering the relationship between variables.
Always.
Which brings us to the final phase of your mission as a Bioforge investigator. We've explored the genetic blueprint, the environmental reality check, the history, but to actually earn your badge, you have to prove it.
You have to make your case. And making a scientific case requires moving beyond simply stating an opinion. Scientific explanations have to be built on evidence.
Because evidence is the accumulated data that supports your claim.
Exactly. And data can take a lot of forms. Direct field observations, data tables, graphs, controlled lab experiments, scientific readings, or massive field studies.
Like the stable isotope analysis of the Sitka spruce.
Exactly like that.
So to structure that data, we use a framework called CER, Claim, Evidence, Reasoning. It's basically the gold standard for communicating a scientific finding.
Let's apply that CER model to the Alaskan tree example right now. Step 1 is the claim. This is a direct statement answering your original question.
So our claim here would be, the environment influenced the growth rate of the Sitka spruce.
That is clear, but it's totally empty without step 2, which is evidence, the hard data.
Right.
For our tree, the evidence statement is, data shows that trees near salmon streams reach a 50-centimeter trunk thickness in 86 years, while those growing away from streams take 300 years to reach the same size.
Okay, I see the value of structuring it this way. But ecosystems are really chaotic. I'm going to push back on step 3, reasoning. Is a formal CER really necessary? I mean, couldn't I just look at a classroom seedling, see that the one with fertilizer is bigger, and just say fertilizer works and call it a day?
Well, no. That is why the reasoning phase is non-negotiable. Without reasoning, you haven't explained the mechanism connecting your evidence to your claim, and you haven't accounted for control variables.
Right, because how do you establish rock-solid reasoning that it was specifically the salmon nitrogen driving the growth and not just, that the soil near the specific rivers naturally gets better sunlight?
Exactly. To isolate the salmon as the key variable, biologists have to compare trees on salmon-bearing rivers against trees on identical nearby rivers that lack salmon runs, ensuring sunlight and rainfall are constant.
So the reasoning is where you bring the underlying scientific principles back into the light.
Yes, for our Sitka spruce CER, the reasoning would be salmon carcasses provide an environmental nutrient, specifically nitrogen, to the forest soil. This environmental factor interacts with the tree's inherited genetic potential, allowing it to accelerate cellular division far beyond the rate of trees lacking that specific marine nutrient.
You tie the environmental condition directly to the biological mechanism of the genetic potential. That totally transforms an observation into a bulletproof scientific explanation.
And that is what a growth investigator does. You don't just notice a result, you explain the interacting causes behind it.
So to officially wrap up your training today and secure your MS-LS1-5 badge, we have 3 Bioforge field note reflection questions for you.
We want you to mull these over to really test your understanding of this lifelong dialogue.
Question 1. Imagine you have a healthy, growing classroom seedling. You move it into a completely dark closet and it quickly stops growing and starts to wither. Is that a genetic factor or an environmental factor at play?
Question 2. Why might 2 dogs of the exact same breed, meaning they share highly similar genetic blueprints, end up being entirely different weights as adults?
And question 3. Think like an ecologist designing an investigation. What specific kind of data would you need to collect to prove that water temperature affects the size of a fish?
And how would you set up that observation to ensure you are testing the environmental condition and not just isolating a unique genetic trait?
If you can answer those 3 questions using the concepts of potential, variables, and evidence, you truly understand the core scientific truth here.
Which is that growth is never driven by just one factor. It is an intricate, continuous combination of genetics and environment.
Because you can now use evidence to explain that interaction, you have officially earned your MS-LS1-5 badge.
Congratulations. It really gives you a fundamentally different lens through which to view the natural world.
It really does. You will never look at a towering redwood or a tiny Arctic plant or even a thriving garden quite the same way again.
Definitely not.
But before we conclude, I want to leave you with one final provocative thought. We spent this deep dive exploring how our internal blueprint and our external environment engage in a lifelong dialogue to shape how we grow.
Right, a dialogue that relies heavily on a certain level of environmental predictability.
Exactly the point. If that dialogue assumes predictable conditions, how might rapidly changing global temperatures chart and shift? I mean, an unprecedented massive environmental shift force organisms to react.
Will plant and animal species be able to trigger hidden epigenetic potential to survive the rising heat and shifting resources?
Or are we about to witness the hard biological limits of their inherited blueprints? It's definitely something to chew on the next time you step outside. Thanks for joining us on this deep dive.